लंग्स, लिवर, क्या आंसर कर रहे हो? Pulmonary angiotensin 1 gets converted into angiotensin 2 when it goes to the pulmonary circulation. Okay, ACE inhibitor सुना है? Angiotensin converting enzyme inhibitors, ACE inhibitors. Okay, so through circulation, when it reaches to the pulmonary circulation, it gets converted into angiotensin 2. Then it is going to affect the flow of the glomerular capillary, okay, and the rate of glomerular filtration. Clear? आपके पास में जो है, आपके पास में जो गर्ल है, खड़े हो बच्चे. What do you mean by GFR? What do you mean by GFR? What do you mean by it? I'm not asking the full form, okay? Glomerular apparatus. Okay, what do you mean by glomerular apparatus? इधर आसपास में कोई, ठीक है ना, वाइवा एक ही को देना होता है. हाँ, लूप ऑफ हेनले कहाँ आ गया बच्चे? ग्लोमेरुलर वहां पे जीएफआर में लूप ऑफ हेनले का कोई रोल है? यस और नो? Sit down. So this is macular densa feedback or tubuloglomerular feedback mechanism. One of the determinants of renal blood flow was through myogenic mechanism. If you remember, due to the change in the entry of the calcium ion, there was a stretch reflex and again a constriction was set. So that was a myogenic mechanism by which we were regulating the diameter of the efferent and the efferent arteriole. Another mechanism for regulating renal blood flow is tubuloglomerular feedback mechanism. Tubulo means we are concerned with the tubular portion of the nephron. Okay, glomerular because it is through this glomerular mechanism, tubuloglomerular feedback mechanism, and this feedback mechanism is coming with the help of macula densa of the DCT. So the total name is tubuloglomerular feedback mechanism. Whenever there is decrease in the GFR, okay, there will be decrease in the distal sodium chloride delivery because 65% of the sodium is already getting reabsorbed at the, क्या हुआ? Okay, maximum of the sodium is getting reabsorbed at PCT segment. Okay, so there will be decrease in the sodium concentration when the filtrate will reach to the distal convoluted tubule segment. Okay, it will, the macula densa will sensitize it that there is problem in the sodium chloride reabsorption. Okay, it will give the feedback through the juxtaglomerular apparatus and there will be correction and there will be decrease in the resistance. When there is decrease in the resistance, that means the diameter of the efferent arteriole will increase. That will result into increase in the renal blood flow. When there is increase in the renal blood flow, definitely there will be increase in the glomerular filtration rate. Clear? फीडबैक हो गया, इसने फीडबैक दिया, सोडियम की कंसंट्रेशन कम हुई फिल्ट्रेट में, इसने फीडबैक दिया, फीडबैक के कारण क्या इफेक्ट आ रहा है? There will be increase in the renal blood flow. When there is increase in the renal blood flow, definitely there will be increase in the glomerular filtration rate. Clear? So renal blood flow is controlled by or auto-regulated by two theories. That is, one is the myogenic mechanism, another is tubuloglomerular feedback mechanism. Clear? If you get a question, you can answer it in a flow chart manner simply. You just have to pinpoint that which part is getting increased or which is getting decreased, what are the stimulating factors and what are the inhibiting factors. Because when we deal with this renal system in detail, the homeostatic mechanism or the pH regulation, okay, then you have a long calculations, but that you cannot write in theory section. Okay, so you have to make a flow chart or tabulate it. Clear? Okay, this is the correction done by the regulation of GFR by angiotensin 2. Another thing was, when there is decrease in the sodium concentration, automatically there will be release or secretion of renin. And what are the effects performed by the angiotensin 2? There is decrease in GFR, decrease in sodium chloride concentration, increase in renin, increase in the angiotensin 2 secretion. But it is only changing the resistance of the efferent arteriole. If you can remember, just remember it, the change brought about by the angiotensin 2 is change in the resistance of the efferent arteriole. Okay, so automatically it will be corrected. If angiotensin 2 is blocked, then it is definitely going to affect the glomerular filtration rate. This is the flow chart you can give very nicely. Okay, if you have time, you can write it down. Decrease in the arterial pressure, that is going to affect the glomerular hydrostatic pressure. That is going to affect GFR. There will be decrease in GFR. Decrease in the sodium concentration at macular denser section. Okay, what will it will do? It will do the decrease in the resistance of the efferent arteriole, plus it will ask the efferent and the efferent specialized cells to release renin. It is going to secrete angiotensin 2. Angiotensin formation, you should know, angiotensinogen is present in inactive form, but get converted into angiotensin 1 and angiotensin 2. Okay, so this conversion takes place, and effect of angiotensin is increase in the efferent arteriole resistance, and that will correct the whole vicious cycle. So are you writing it, or shall I move it to the next slide? राइटिंग और कॉपी तो मुझे दिख नहीं रही है. मेंटल other factors that influence GFR. Okay, fever or pyrogens. Okay, what they do, they increase in the glomerular filtration rate. Glucocorticoid increases GFR. Aging, with age there is decrease in the tense-peripheral functional ability of the nephrons. Hyperglycemia, that is increases GFR. Example, diabetes mellitus. Dietary protein, if you take a high protein diet, okay, then there will be increase in the GFR, because approximately 20 to 30% increase in GFR is seen if a person goes for high protein diet. Okay, so these are the factors which are going to affect the. So this is the story of protein ingestion. Okay, if excess of protein is taken in the diet, there will be increase in the concentration of amino acids. If there is increase, the amino acid will be absorbed. More than 90% of this will get absorbed or reabsorbed in the PCT segment. Okay, whenever amino acid are getting reabsorbed, along with the amino acid, what is getting reabsorbed? सोडियम. क्लियर? जस्ट रिमेंबर, अभी नेक्स्ट उसका पावर पॉइंट यही है कि इन सब का रीएब्जॉर्प्शन किस मैकेनिज्म से होता है, बट अलोंग विद द अमाइनो एसिड व्हाट इज गेटिंग रीएब्जॉर्ब्ड इज सोडियम. सो प्रॉक्सिमल ट्यूबुलर सोडियम रीएब्जॉर्प्शन इंक्रीजेस. If there is increase in the sodium reabsorption at the PCT segment, there will be decrease in the sodium concentration at the DCT segment. So if there is decrease in the DCT segment, macula densa will give feedback mechanism to increase the renal blood flow. If there is increase in the renal blood flow, automatically there will be increase in the glomerular filtration rate. Okay, this is the story of there is increase in the diet or protein intake. Okay. very subjective topic, okay? Just try to focus it, remember it through diagrams, okay? We'll have diagrams, representation of each and every section of the nephron, and we'll make it a list or a list which things are getting reabsorbed at which particular segment. Clear? So it will be very much clear for you. When we are dealing the glomerular capillaries, the colloidal, the oncotic pressure of blood is how much? नींद आ रही है इसको? कितनी होती है oncotic pressure blood की? खड़े हो जाओ. This is the scenario of a morning class, 9 to 10. Okay? Till now, this picture might be clear with you what is reabsorption and what is secretion. Reabsorption, when the substance from the filtrate through the epithelium lining of the tubule enters into the interstitial space. From the interstitial space, it has to enter into the peritubular capillaries. Okay. Once it enters into the peritubular capillaries, this is known as tubular reabsorption. When a substance like urea, it has to be secreted. So from the peritubular capillaries, okay, the urea or the drug toxins or any other product which has to be excreted, okay, it will come from the peritubular capillaries into the interstitial space. From the interstitial space, it will enter through the basolateral membrane of the tubular epithelium cell, enter into the cell or through the transcellular junction, and it will go into the tubular lumen. So this is known as tubular secretion. Okay. Now the term will become tubular reabsorption or tubular secretion. Okay. So whatever the amount of the concentrate of the filtrate. Okay. Filtrate के अंदर अभी मैंने क्या क्या बताया था? Glucose, amino acids, some part of the proteins, maximum is water, sodium, potassium, calcium, lactic acid, bicarbonate. These are the substances which are present in the filtrate along with water. Of this composition, the oncotic pressure is same as that of blood, that is 300 milliosmoles. Out of this, 65% of the substances are getting reabsorbed at proximal convoluted tubule. This much reabsorption has to be done. So histological picture of PCT is, it has number of villi. Number of villi, what it will do? It will increase the surface area for reabsorption. Powerhouse of the cell क्या होता है? खड़े होके दे दे भाई, थोड़ी नींद भागेगी. हाँ, माइटोकॉन्ड्रिया. तो पीसीटी के सेल्स का क्या फंक्शन है? फंक्शन क्या होता है? क्या होता है? वापस बोलो. अभी मैं इतनी देर से क्या बोल रही हूँ? रीएब्जॉर्प्शन. Okay? When we are looking for the answers, we look for keywords. So it is tubular reabsorption. Okay. So if this 65% of the tubular reabsorption has to be done, so mitochondria will be more in these cells. Clear? ठीक है तबियत? Better. Calculation of tubular reabsorption when excretion substance of the to be excreted is less than the filtration. So these calculations very rarely, बहुत remotely they can be asked. Okay. We will come main for the reabsorption and water solutes. Okay. Different solutes will work differently. So most important is water, okay, and other solutes. These are the different mechanism of reabsorption. This is the picture. This is the peritubular capillary. These are the tubular cells, okay, cells which line the lumen, and this is the lumen, okay. Filtration which is entering into the lumen. In between the two cells is the paracellular pathway. through the cell, it is transcellular pathway. Okay. Sodium, potassium, ATPase pump, they are present on the lateral basolateral membrane. Lateral side पे होते हैं, they are not present at the luminal surface. Okay. Osmosis, through osmosis, movement of water takes place. But this osmosis, okay, the ion which is responsible for the osmotic movement of the water is sodium. Okay. So the sodium and the water is obliging it. So this type of water reabsorption taking in the PCT is known as obligatory type of water reabsorption. You have two type of water reabsorption in the nephron. One is the obligatory type of water reabsorption and the other is the facultative type of water reabsorption. Facultative type of water reabsorption takes place in the presence of ADH at the collecting duct. PCT, water reabsorption takes place due to the movement of sodium ion, and this type of water movement takes place by which mechanism? Through osmosis, and this reabsorption mechanism is known as obligatory type of water reabsorption. Clear? हाँ या ना? इतना okay, primary active transport mechanism by this sodium potassium ATPase pump. Okay, side junctions. Okay, now the most important is when there is presence of sodium potassium ATPase pump at the lateral surface. Okay, the filtrate is having enormous amount of the sodium. Okay, that sodium has to be reabsorbed. Okay, so what is the mechanism that is involving in this type of reabsorption is secondary active transport mechanism, but the primary cause is the functioning of sodium potassium ATPase pump. So what sodium potassium ATPase pump is doing, there is increase in the sodium concentration inside the cell, so it is moving sodium out and taking potassium in. When this type of movement takes place, there will be decrease in the concentration of sodium inside the cell. So it is creating what type of gradient? Electrochemical gradient. एक तो ये पिक्चर हो गई कि ATPase पंप क्या कर रहा है? सोडियम का फ्लक्स कर रहा है. Sodium is moving out. When sodium is moving out, sodium concentration inside the cell is decreasing. Another picture is the filtrate which is coming from the glomerular capillaries. The sodium load is enormous. So there is electrochemical gradient which is created from the lumen through the cell. Okay. Now at this junction, luminal side, we have a channels of proteins which are responsible for carrying sodium from the filtrate into the cell. Clear? Special channels of proteins which are present, and they will carry the sodium from the filtrate into the cell. But वो तब होगा जब वो electrochemical gradient create होगा. और electrochemical gradient किस वजह से create हो रहा है? Because of the functioning of the sodium potassium ATPase pump. So if there is problem in the sodium potassium ATPase pump, there will be problem in the electrochemical gradient. So there will be no proper reabsorption of sodium through the secondary active transport mechanism. When we are talking of secondary active transport mechanism, there are channels of proteins which are present at the luminal side. Okay, and they will bind or they will combine with certain ions, okay, or substances, and through this electrochemical gradient, they will help these substances to move inside the cell. Once they are inside the cell, the forces will act or responsible for their movement into the peritubular capillary. Clear? So this mechanism has to be very much clear how the secondary active transport mechanism takes place due to the functioning of the primary active transport mechanism, which is responsible for creating the electrochemical gradient. कहाँ के बीच में? Lumen and cell. And what we have in lumen is the filtrate. Clear? and the oncotic pressure of the lumen filtrate at PCT is 300 milliosmoles, similar to that of the oncotic pressure of the blood. Okay, so basic, when we take a section of the PCT, we have to remember three basic things. First is oncotic pressure. Second, the substances which are getting reabsorbed. Third, the substances which are getting secreted. So the picture will be clear when we take a cross section. Okay, in few slides I'll show you the section, so it will be easy for you to remember. So you get MCQ, that which substance is getting reabsorbed at which segment of the nephron. Okay, except this. So it will be clear. Okay. So this is the example of secondary active transport mechanism. This is the example of symport, and this is the example of antiport transport mechanism. When we are talking of glucose transport mechanism, this is the carrier is known as sodium dependent glucose transporter. They have two types, glucose transporter 1 and glucose transporter 2. In previous slides, we were talking about the water reabsorption. So water reabsorption, we have proteins which are present on the luminal surface. They are known as aquaporins. So we have 4 types of aquaporins, and 2 types are present at this PCT segment, and 2 types they are present at the collecting segment, but they are getting influenced by ADH. So ADH पे जब बात करेंगे तब अपन क्या बात करेंगे? Facultative type of water reabsorption. Here we are talking of obligatory type of water reabsorption. Now in case of glucose, okay, खड़े हो? इतने तो जगे हुए हो कि सुन ही लोगे, खड़ा होना है, बैठना है. Does glucose appear in urine? Yes. फिजियोलॉजी की बात कर रही हूँ मैं अभी, मैंने कोई पैथोलॉजिकल कंडीशन तो बोली ही नहीं है. Normally it is not present. Still, if we increase the amount or the concentration of glucose, will it appear in urine? Under normal physiological condition? No. What is the mechanism? Why it doesn't appear? बताओ. Secondary active transport mechanism. Okay. This, बैठो. Secondary active transport mechanism. Okay. They are present in all the nephrons. Clear? But this is being done with the help of channels or carrier proteins. Okay. So they have a threshold, or their number is to a particular limit. Okay. So that is the maximum glucose that can be reabsorbed from the filtrate is 375 milligram per minute. Okay. That is known as transport maximum. But still what happens is, if the glucose concentration or load increases above 180, it starts appearing in the urine. Clear? The number of proteins which are responsible for transporting sodium dependent glucose transporter 1 and 2. Okay. They have a limited number present on the luminal side of, we are talking of a single nephron, but we have millions of nephrons, and each nephron is having these type of channels present. Okay. But the functional ability of each nephron is not up to 100%. Clear? So there is appearance of glucose in the urine, still the concentration is up to only 200 or 180 milligrams. So this difference in the theoretical and practical aspect, okay, that is known as splay. So you get a short note on splay, S-P-L-A-Y. So what do you mean by splay? That is the transport maximum. The capacity should be theoretically 375 milligrams, but still after 180 start appearing in the urine. So this difference in the reabsorption capacity, though the threshold and the transport maximum Okay, so this variation is one of the reason given in books is that all the nephrons might not be functioning total 100%. Clear? So which is resulting into this deviation, and this deviation of reabsorption of glucose is known as splay, SPLAY. Just go and see in your book क्या ग्राफ दे रखा है स्प्ले का. Okay, so just write down splay. Some substances have a maximum rate of tubular transport due to saturation of carriers or limited ATP, etc. Once the transport maximum is reached for all nephrons, further increase in tubular load are not reabsorbed and they are excreted. Okay. Threshold, what is threshold? It is the tubular load at which transport maximum is exceeded in some nephrons. This is not exactly the same as transport maximum of the whole kidney, because some nephrons have lower transport maximum than others. Okay, so all the nephrons, example is glucose, amino acid, phosphate and sulfate. Clear? So when it is the story of glucose, the term given is splay, and you will get a graph. Ideally it should go straight line, but it will be deviating and it will appear into the urine. Okay. So reabsorption coupled with sodium reabsorption. So due to this movement of sodium, okay, continuous movement of sodium, glucose, amino acid, so what will happen? There will be increase in the concentration of chloride. Okay, बाकी ions क्या होंगे, वो filtrate में ज़्यादा हो जाएंगे. ठीक है. So this will create a difference gradient, okay, and they would like to follow the paracellular pathway. What pathway they will try to follow? That is the paracellular pathway. Okay, so it is easy for us to remember that sodium is responsible for what type of reabsorption? Sodium is responsible for water, sodium is responsible for glucose, sodium is responsible for amino acid, sodium is responsible for lactic acid. Clear? It is responsible for the movement of lactic acid. Okay. Bicarbonate is also getting reabsorbed, but that is also something different. The bicarbonate which is coming into the filtrate, जो आयन फिल्ट्रेट में आ रहा है वो as such reabsorb नहीं हो रहा है, it is getting changed. First it will combine with carbonic anhydrase and result into the carbonic acid, which will dissociate into water and carbon dioxide. Another story will take place in the epithelium cells where water and carbon dioxide, okay, so they will combine and for carbonic acid and they will dissociate into hydrogen ion and bicarbonate ion. अपन क्या देख रहे हैं, सोडियम के साथ में हाइड्रोजन क्या हो रहा है, सिक्रीट हो रहा है. तो हाइड्रोजन आयन कहाँ से सिक्रीट हो रहा है, सेल से. हाइड्रोजन आयन की फॉर्मेशन कैसे हो रही है, कॉम्बिनेशन ऑफ वाटर एंड कार्बन डाइऑक्साइड इनसाइड द सेल इन द प्रेजेंस ऑफ कार्बनिक एनहाइड्रेस, डिसोसिएशन ऑफ हाइड्रोजन एंड bicarbonate ion. Bicarbonate, we have to reabsorb hydrogen, we have to eliminate or secrete or excrete. Okay. Urea, okay, it can easily move through the transcellular pathway. Water also through osmosis or easily it can move with. So the mechanism of coupling water, chloride, urea reabsorption and sodium reabsorption, and this is the flow chart which will make it very clear. Along with sodium, water reabsorption, which type of obligatory type of water reabsorption? Okay, lumen will get negatively potential, increase in the chloride concentration, increase in the urea concentration, passive type of urea reabsorption and passive chloride reabsorption. This is the picture I was talking about. Histological isoosmotic is the concentration, same as that of blood. These are the substances which are getting reabsorbed, okay, and these are the substances which are getting secreted. Okay, so PCT segment in one slide, it is very much clear that which substances are getting reabsorbed and which substances are getting secreted. And whatever the amount, okay, whatever the quantity. 65% of the reabsorption takes place in PCT. Please remember it. Okay, how much percentage? 65% is getting reabsorbed in the PCT segment. क्या टाइम हो गया? सबकी घड़ी अलग है क्या? 45 तो मुझे इसमें दिख रहे हैं. So this is the graph which can be seen changes in concentration in proximal tubule of different substances. Proximal tubule में अमोनिया, अमीनो एसिड, ग्लूकोज, इसकी कंसंट्रेशन कम हो रही है, बाइकार्बोनेट रीएब्सॉर्ब हो रहा है, उसकी कम हो रही है, क्रिएटिनिन, यूरिया की बढ़ रही है. सिंपल, पर आपको एक स्लाइड के अंदर हो गया. ठीक है, नेक्स्ट अपन कवर करेंगे इसके डिफरेंट सेगमेंट एंड अलोंग विद दिस विल डू अबाउट द होम्योस्टेसिस फंक्शन ऑफ किडनीज. एक पेपर देना.